It's got a little red light on it. Okay. Okay, thanks very much, and also thanks very much for inviting me to talk here. So I'm going to talk about something maybe a little bit different from what a lot of this workshop has been about, and I'm going to talk very much about problems in population genetics, which is broadly trying to understand patterns of genetic variation. And I want to motivate this by thinking about where we are as a community in terms of the data available for looking at human genetic variation. We have to admit we're in a sort of a pretty amazing position of having vast amounts of information on really quite large numbers of individuals from things like the HapMap project. And as Richard talked about earlier in the week, we're very shortly going to be in a position of having even more data on an even more ridiculous scale from the 1,000 Genomes project. So we have a genome's worth of data on genetic variation, and this is sort of a snapshot of what it looks like. This is data taken from the HapMap project. One of these is probably a pointer. Aha, thank you. So this is data from the HapMap project, and let me just talk you through what we're looking at here. This is about maybe a few hundred kilobases on chromosome 17, I think. We're looking at polymorphisms, single nucleotide polymorphisms that have been genotyped in the study. Here are the genes in the region, and here's the data. Now, what we're looking at here is each row along here is a chromosome, and each column is a single nucleotide polymorphism. And just for visual clarity, rather than show you the A's, T's, G's, and C's, what's been done is to arbitrarily color one of each allele as yellow and one as blue. Now remember, we're only looking at biallelic polymorphisms here, so that's why we can get away with just having two colors. So we have, this is data just from one of the populations in HapMap. This is just a small part of the genome. We have obviously much more data than this, and we'd like to use these data to learn about some of the most fundamental molecular and evolutionary processes that are known to have influenced human history. Things like, so fundamental molecular processes like recombination or mutation and so on, evolutionary forces like natural selection, and also human history, how have people migrated across the world and bred with each other over time. So that's the sort of the setting. I want to give a couple of more specific examples of what we're trying to do. Here's one. We want to learn about the process of recombination. Now, we know that in humans, the rate of recombination, which is this process that shuffles up variation between generations, varies along a chromosome. But there's remarkably little that's known about what determines where these recombination events occur, and we'd like to learn about those processes. We can actually learn about recombination from patterns of genetic variation, and one of the things I'll talk about at length is how we do this. And so we'd like to take data that looks like this again. This is just I can't remember which part of the genome this is. We'd like to take the data that looks like this and to learn about the recombination profile across this interval, and then take that profile across the whole genome and use it to learn about the processes that determine why recombination occurs in particular places. And just if we were to give the game away for this region, what we actually learn about recombination from studying this region is that there's a recombination hotspot, i.e. a region at which recombination is incredibly concentrated, slap bang in the middle of this region. And it's not totally obvious that if you just stared at the data, you'd see that. And so we'd like to develop ways of making these inferences. So that's one kind of setting. Here's another one. I'm going to spend quite a lot of time talking about how we think about genetic variation. And a key point will be that the genealogical relationships, or which I'll describe in a bit more detail in a second, between sequences are very informative about the underlying process. Or put another way, if I told you the tree at any position on a genome, or I told you lots of trees in lots of different places, then those trees would be very informative about underlying processes. So we'd actually like to know what the genealogy is of the human species, without obviously trying to go back through records to try and do that. So we'd like to, rather than sort of go back through parish records, which obviously only go back a small period of time, we'd like to estimate these relationships from DNA sequences, reconstruct history going back hundreds of thousands and even millions of years. And then we could use these estimated relationships to learn about diverse processes. We could use them to learn about human history, demographic history primarily. We could use them to learn about natural selection and how that's varied across the genome and across different parts of the world. And actually, there are some very applied uses of estimating these genealogies. For example, we can use them to aid the fine mapping or even the primary location of disease-associated mutations. So again, just to explain what I'm talking about, here's a set of genetic variation again, and I'd like to be able to point at a position along the genome and say something about what the relationship, the tree relating chromosomes to each other looks like at that position. Okay, so those are a couple of tasks that I would like to be able to do when faced with a genome's worth of genetic variation. So how am I going to go about achieving these tasks? Well, a natural place to start is to think about what's a suitable way of modeling genetic variation. Can I come up with a simple, sensible, probabilistic framework for exploring trying to make these inferences? What's a natural model for variation, and how can I use that model to drive inferences about these underlying processes? Now, we actually have an extremely beautiful and quite well studied probabilistic model that does a remarkably good job of describing patterns of genetic variation and the way in which diverse processes influence variation. And it's called coalescent modeling. And the key insight in coalescent modeling is that the right way to think about patterns of genetic variation, whether it's the chromosomes in humans or diversity in HIV, is to think about the relationship between the sequences that you've got. Okay, to think about this underlying genealogy. And the coalescent model itself describes the distribution of these trees or these genealogies, which you get between sequence sampled from idealized populations. And then you can reduce the degree of idealism in various ways. And so it's primarily a description of the genealogies, but of course what we see are patterns of genetic variation. And so how do you go from genealogies to variation? Well, what you do is you can map mutations onto these genealogies, and that results in the variation we see. So here's a more detailed example of this. Here's a genealogy, I think for about 20 sequences. This is actually a simulation. This is a draw from the coalescent process. You can see it's got a sort of fairly characteristic shape. And we can think about the patterns of genetic variation that might have given us. So again, each row is a chromosome, each column is a mutation. And the variants that we see over here are the result of mutations occurring on this tree. So if I put a mutation on this branch, it will be shared by those two guys there. Maybe it's that mutation there. Okay, you can see that mutation is shared by these guys. And you can see that the variation I've got there looks maybe a little bit like the pattern of variation that I put up to start with, which was truly empirical. Okay, so I just wanted to give you, for those of you who haven't come across coalescent theory before or have but don't quite know where it comes from. I just wanted to give you a small insight into where it comes from. So where do these trees actually come from? Well, what we can think of modeling genetic variation in human populations by a sort of a very simple beans in a bag kind of model, where we have a population, which here is represented as just a bunch of blobs. Each of you are a bunch of blobs. And these blobs reproduce in a series of discrete generations, where every generation, the number of offspring that a blob leaves is a binomial distribution, is binomially distributed. And so some blobs are unlucky and leave 0 offspring. Other blobs are lucky and leave multiple offspring. And so what you see over time is you can follow these sort of lines of ancestry through this population. And this process goes on and on and on forever, essentially. And so when we look at individuals in the present day, which are at the right-hand side of this picture, in terms of events that could have influenced the patterns of variation we see in today's population, we can actually throw away a lot of this history. We don't need to care about those individuals in the past who have left no descendants in the present day. And so the relationships, genealogical relationships between these blobs is described as a tree, whereby you start with your n individuals, and then looking backwards in time you have a monotonically decreasing number of ancestors, and you're guaranteed to get back to a single ancestor at some point in the past. And it's mutations that occur in these transmission events that obviously lead to variation in the final population. Now there's a further and actually very important modification to this, is that of course when we do our experiment we don't sequence everyone in the entirety of the world. We only take a sample, here represented by these blue blobs, and so the relationships between these blue blobs are also described by a tree that is embedded in this bigger population. And so the coalescent is a model for these genealogies of samples, finite samples taken from actually infinitely sized populations. It's a very simple model. It has a very simple probabilistic structure. It's easy to simulate from, and that's partly why it's become such an important tool in the analysis of genetic variation. Sort of most importantly, rather than representing diversity by this sort of full, very complex history of individuals reproducing and so on, we're going to strip most of that away and just follow these lines of descent, and that's going to give us our tree. And so coalescent modeling allows us to describe the distribution of genealogies of samples from from idealized populations. And just to give you a sense of the kind of inherent variation, here are 6 draws from the coalescent process with about 10 sequences, and what you can see is that they're sort of quite varied in their shape. For example, we've got some that are very deep, some that are very short. Quite often you see these sort of deep partitions of data of lineages into 2 parts of a tree, and so on. And so we can build up an understanding of these trees through simulation. These trees are obviously high dimensional, so it's going to be hard to represent the full distribution in any sensible way. So that's modeling the genealogies, and of course we can use those to describe the distribution of data that comes out of this process at the other end. So again, I've just thrown mutations down onto those trees to generate the patterns of variation I've got here. So that's the very simplest version of coalescent modeling, in which there's no recombination. There's just a single tree that describes the history of a set of sequences. Clearly we want to generalize the model to allow us to include lots of different processes. In many settings, the most important one, and actually the one I'm really going to focus on here, is recombination. And when you've got recombination, the key difference from what I've just described is, rather than there being a single tree that describes the relationships between samples, there's actually a much more complex graph that describes the joint distribution of trees along the genome. So put another way, when there's recombination, the tree that I get at position X will be different from the tree I get at position Y. If there's not much recombination between X and Y, they'll look pretty similar, but if there's lots of recombination, they'll look very different. And so I can jointly describe this set of genealogies using an extension of the coalescent model, which is called the ancestral recombination graph. And here's an example of an ancestral recombination graph in the simplest possible case of two sequences. Sorry, just going to have to blow my nose. So if you take this picture down here, it looks a bit like a tree, but actually you can see there are sort of two sets of lines here. If you just focused on the blue part, you can see that there is in fact a well-behaved binary tree, and there's a mutation here on this tree that means that the two lineages here will share that mutation. Likewise, if you just focus on the red part, you can see that there's a well-behaved tree, and that there's a mutation here which will be shared by these two guys here. Now the red and the blue trees are themselves sort of linked, such that the red tree and the blue tree look pretty similar, but at this point here there's a sort of a bifurcation in the sense that the two trees take slightly different paths, and the key point here is that this is an ancestral recombination event. So if I take my gene, say I take one of my copies of my gene that either came from my mum or my dad. Suppose it came from my mum. That gene itself might have been a mosaic of the copies that she got from her mum and dad. And so if I look to the left and the right of the break point, in that mosaic structure, then I would see this kind of graph. Okay, so this is clearly a very, very simple graph, and for any moderate amount of recombination or any moderate sample size, you typically end up with very complicated graphs which are very hard to show visually. Nevertheless, I think the key point then is if we have a set of genetic variation data that looks something like this, then the right way to think about these data is that at any one position along this region, there is an underlying tree. For example, here's the tree at the start, there's the tree at the end, but those trees can change as you move along the sequence. In fact, what's happening here is that we've got the same tree, same tree, same tree, and then it suddenly shifts because in the middle of this region there's a recombination hotspot. Nevertheless, there is some correlation between the two trees either side of that hotspot. So if I looked at the sort of the fine details of how individuals are related to each other, I'd find some pairs of individuals or small clades down here, which I'd also see in the point over here. So these trees are not they're not like independent draws from the process. They are correlated in very important ways. Okay, so what we have now then is a probabilistic model, which we're broadly referred to as the coalescent, which allows us to describe patterns of genetic variation under diverse processes. When I say it allows us to describe, really the most clear way in which that occurs is through a simulation process, i.e. we can describe a distribution by looking at draws from that distribution. But because of the high dimensionality of these args and the data that comes out of it, it's impossible to, it's not like a normal distribution. It's very hard to describe in any full way. Okay, so that's essentially the background to coalescent theory. Now, okay, so that's what the model looks like. What about performing inference under the model? So broadly, we'd like to use this model, or the theory generally, to drive inference about these underlying processes. No-brainer. More specifically, or I've written generally there, we'd actually like to calculate the likelihood function. That is, for a given data set, we'd like to be able to calculate what's the probability of observing that data given a set of underlying parameters. And we might say want to maximize that or perform some kind of Bayesian inference. However, there is a problem, which is that there's a many-to-one mapping of these graphs, or genealogies, joint genealogies, to the data. That is, if I give you a data set, there's not a unique tree which could have given you that data. There are lots of different trees, lots of different joint trees. And so to calculate this likelihood, what I've got to do is to integrate out the missing data, which is the genealogy. So basically, I'd have to consider all those trees or genealogies which are compatible with the data or could have given me the data. I can integrate over that, and that will give me the likelihood. Unfortunately, that cannot be done analytically. The space of these args is simply too large, and so basically in anything but the most trivial example, you have to use Monte Carlo techniques to do this integration. So just to give you a sense of how you can do this integration, here's a very simple data set. I have a tendency to represent data in lots of different ways. Here, rather than blue and yellow squares, what we're looking at are lines representing chromosomes and red and blue blobs representing the mutations that have occurred in the history and lead to variation in the sample that I've collected. Okay, and what I want to do is to calculate essentially what's the probability of observing these data. And so what I want to do is to think about all the histories, these underlying args, that could have given me these data in the first place. So what I can imagine doing is, in fact, taking these data and proposing a history that is compatible with what I've got. And here is just one such history in which, looking backwards in time, the first thing was a coalescent event between these two, then had a recombination event, a coalescent, a mutation, and so on. And this history gives rise to these data. That's a relatively simple history where there's a single recombination event. And to calculate the probability of the data given that, well, just in this particular setting, because the coalescent process has a particularly nice Markovian structure, you can just multiply the probability of these events together to get the probability of the joint probability of the history and the data. And what I would do is to essentially try and sum over all possible histories, or more specifically, pick out histories which are highly likely to have contributed to my data. And I'd have to use techniques like particularly importance sampling to try and do that in an efficient manner.
Is there a need to consider genotyping errors?
Okay, so everything I'm going to say for the moment doesn't consider genotyping errors. We're going to focus on the simplest case, and then we'll try and sort of put extra things in later. At the moment, we don't explicitly consider genotyping errors anywhere, but the model that we do fit allows for recurrent mutation on the genealogy, which is a bit like genotyping error. Richard?
So given the shape, you can integrate over genotyping errors and multiple mutations.
Yes. Okay, so of course I could just use a pruning algorithm to calculate the likelihood, the probability of these data, or whatever, given this set of marginal genealogies. That isn't a complicating factor in any of this. The complicating factor is efficiently exploring the space of these graphs.
The events are mutations and splits.
Yes, all of these things on the left-hand side are what I would call an event, a coalescent, a recombination, and a mutation. They're all my events. And the theta down here is just sort of a generalized set of parameters. Okay, so that's ideally what we'd like to do, we can't do it perfectly, we could come up with Monte Carlo strategies for doing this in some way. However, there still remains a problem, which is that efficient exploration of the space of args is a difficult problem, particularly for large data sets, where large I mean anything more than about 10 sequences and a few SNPs. So currently, the difficulties of performing efficient, exact, or even semi-exact genealogical inference within a coalescent framework currently seem insurmountable. We simply cannot fit the coalescent model, even with certain approximations, to large data sets. It's just not possible. So we had the quote yesterday that all models are wrong and some are useful. I think in this case all models are wrong, but this one's useless in the sense that we actually can't do anything with it really. Okay. So what can we do apart from give up and go home? Well, there are a bunch of things that we might try to do. One area that's received quite a lot of attention is what you might refer to as dimension reduction, which is rather than deal with the probability of observing your data, you might deal with the probability of observing some summaries of your data. And there's a field called Approximate Bayesian computation, which essentially tries to do this. That's one approach. Another approach, which it's possible Richard talked about, but I don't know for certain, is that rather than deal with the coalescent, you can try and take out the best bits of the coalescent and come up with new models which allow for efficient inference, which as I say, captures some of the key features of the coalescent, but throw away a lot of the complexities. That's a nice idea and has proved very useful in some settings. And a third possibility, which is what I'm going to talk about, is that you can, rather than approximate the model, you can approximate the likelihood function. And the approach I'm particularly going to talk about that has proved useful in a bunch of different settings is to, rather than look at the whole data at once, is to break the data up into little bits, take subsets of the data for which you can compute the likelihood function, and then combine those in some sensible way. And this is an area that's broadly referred to as composite likelihood, and it is fairly widely used in statistics. But as you'll see in a little bit, while it's a nice idea, it does come with certain complexities. Okay, so I'm going to talk about two uses of composite likelihood in the analysis of genetic variation, and the first example is estimating the recombination rate. So I said we can't calculate the likelihood for a complex data set consisting of many sequences and many SNPs. That's true, but if I gave you a much smaller part of the data, which was in fact just 2 SNPs, you could compute the likelihood function in using these Monte Carlo techniques in a reasonable time. So the idea that I'm going to explore, originally proposed by Dick Hudson a few years ago, is to say we can't compute the likelihood. We can do it for the whole thing. We can do it for these subsets of the data. So let's simply take the likelihood surfaces from these subsets and multiply these marginal likelihoods together. Okay, so it's a naughty thing to do. You're treating these subsets of the data as independent when they're clearly not independent. But if it leads to a useful method of inference, you might be forgiven. So how does the method do? You'll see in a second, but I mean, to cut a long story short, it does do remarkably well in terms of point estimation. So here's just a cartoon of what I'm going to do. Here's a data set consisting of about 30 sequences. Each of these is a haplotype. That's the multiplicity of the haplotype. And there's the pattern variation that we see within this. What I can do is to take pairs of SNPs, and I could imagine taking all pairs of SNPs, or I could select different pairs of SNPs to focus on. Initially, I'm going to just take all pairs of SNPs. And for each pair, I'm going to pretend that the rest of the data isn't there. And then I'm going to use these Monte Carlo methods to estimate the the likelihood surface for the underlying recombination rate. Okay, so big R is just here meant to mean the recombination rate. I can do that for those two SNPs. I can do it for these SNPs, and I get a different surface. Okay, and then to get the approximate surface for the complete data, then I'm just going to add these log likelihood surfaces together in a sensible way. Obviously, I've got to scale the distance between SNPs in a sensible manner. And so this red line shows you the curve that I get for these particular data. Okay, and it has a peak, and you might imagine taking that, the point where that maximizes, as a sensible estimate of the recombination rate. Now, actually, this data set is small enough that if you really, really push it, you can get full inference to work on it, although you have to run the computer for a couple of months. And the blue line here is what you get, obviously rescaled, if you use the full likelihood, if you do this full likelihood. So what you can see in this particular example is that the composite likelihood approximation basically gives you the same point estimate of the recombination rate, but it is a much more sharply peaked surface compared to the true likelihood.
Am I right that this is essentially the analog of BSAG's pseudo-likelihood?
Yes, that's right. Yep.
Out of curiosity, is this simulated data? I mean, do you...
This is simulated data.
So do you know what the function that went into it was?
Yeah, so the truth is 3, actually. Yeah.
The truth is 3, and it was just a flat 3, as it were.
It's a flat 3 in this case, yes. Yes. You can't get these full likelihood methods to work for variable rate.
Okay.
Yeah, just fall over. Won't do it. Okay. And this is generally what you see if you do a whole bunch of simulations in this space where you can just about get the full likelihood methods to work. You typically see very good correlation in terms of the point estimate, but the composite likelihoods are over certain because they're going to double count information. You know, if I'm going to include all pairs of SNPs in this, they're not independent. There's a dependency structure which I'm ignoring, and that leads to me thinking that I'm more certain about what the true value is than I should be, and that, you know, in the composite likelihood, I'm actually throwing a lot of data away, so this really should be flatter. But you can see it's more peaky. And that's a typical feature of composite likelihood. So what are some good things and bad things about composite likelihood? Good things: estimation using the composite likelihood can be made extremely efficient. In particular, you can use tricks like making lookup tables and doing various interpolations that allow you to analyze very large data sets, in fact whole genome data sets, reasonably efficiently. It performs very well in simulations in terms of point estimation, and as I'll describe in a second, it generalizes nicely to deal with variable recombination rates. Those are good things. The bad things: it throws away a lot of information. We're only looking at pairs of sites. We're not going to look at the whole thing. I think what's remarkable is how good the estimates are, even though you're throwing away a lot of data. So that's one bad thing. A worse thing is that it's actually not a true likelihood. It's just a function which I'm maximizing. And specifically, it typically underestimates the uncertainty, or it's over certain essentially, because of this double counting. Now, as I'll say a few words about this later. This is bad, but it's maybe not as bad as you might first think, because you can actually bound how bad that is going to be. And so you can actually try to correct for this double counting in certain places. And that's actually, that's not an area I've looked at hugely. I've done it, I have looked at it in this case of recombination rate estimation, and we have a bound which does lead to useful, to actually reasonably good estimates of uncertainty. Okay, so basically you can put another way, you can take this surface and you can do a power transformation of it. And the question is what power to transform. That's not clear, but you can bound what the worst you should do is and use that. And that seems to work reasonably well. Okay. Right, so that's fitting a constant rate. What about fitting a variable recombination rate? As I said at the beginning, we know that recombination in humans is not uniform. It sharply peaks in various places. We'd like to generally estimate what the profile of recombination is. And so this is a specific example of a sort of more general problem of how do you estimate a process where there's a variable underlying rate in which you don't want to make a priori statements about where those changes might be, or indeed how many changes there are. And so there's a useful technique, at least in the Bayesian setting, for estimating variable rates over intervals using a technique called reversible jump MCMC that was developed by Peter Green a little over 10 years ago. Many of you are probably familiar with it, but for those of you who aren't, I just want to sort of describe what you're trying to do. Here's the region I'm interested in, and the red arrows are meant to illustrate where I've got SNPs. And I'm going to start with some guess at what the recombination profile looks like across the region. And I've just color-coded it so that blue means low recombination rate, red means high recombination, and pink means something in the middle. So what I can do is to start out with an initial guess, and I can explore what the reversible jump technology allows you to do is to explore the posterior distribution of these piecewise constant maps in a Bayesian framework. And you do that by setting up a Markov chain, which basically takes your current guess and proposes various subtle changes to it. For example, I might take a region that I've got here as piecewise a single region, and I might split it into two. Or I might take two blocks and merge them into one. Or I might change the size of a block, or I might change the block rate, so increase the recombination rate in there. It's fairly simple. There's some accounting you have to be quite careful about. But if you do it reasonably well, and it's a well-established technology, I have no claims to any novelty here. Basically, it's just like Metropolis Hastings, where this psi prime is your proposed map, sized existing map, and your acceptance rate, alpha, is given by the minimum of what looks like the basic Metropolis Hastings ratio, the ratio of the likelihoods, or here, of course, I'm going to use the composite likelihoods, the Hastings ratio, the ratio of the priors, and this is where the careful accounting comes in. You've got to make sure that you essentially keep the spaces of the two places the same. And we include a prior on the number of change points that encourages smoothing. So it allows it says we don't want to have too many change points. And so this is what it looks like. This is a region of the HLA, and here's these are just samples from this pseudo posterior. So it thinks that there are sort of generally three peaks in recombination over here, probably two here. There's one over here with possibly something else going on over there. So you run your chain for a reasonable amount of time, and then you can summarize the output of it, for example, by just looking at the mean posterior estimate at every point. But of course, remember, this is not a true posterior. This is a pseudo posterior. Of course, we need to validate this methodology. So how can we actually convince ourselves that the estimates that we get out of this are sensible? Well, very fortunately, we have two sources of information that we can compare our results to. One is that in some regions, and in particular in this region that I showed you the samples from, Alec Jeffreys in Leicester has done very fine-scale estimation of the crossover rates within this region, not by studying pedigrees or anything, but actually by looking at crossover events in sperm. So you can build up very fine-scale genetic maps by looking at crossover events in sperm. These are very difficult experiments. You can't do them on a large scale, but you can do them for some regions. And these experiments allow us to validate the methodology in particular regions. And just to cut a long story short, so here's a summary of that, the rate estimates within that region, so on a log scale. So generally it thinks there are 3 hotspots over there, 2 in the middle, and 1 on the right. And this is a parsing that actually calls hotspots using a slightly different methodology that I haven't explained. So that's what we estimate, and the red lines are what Alec Jeffreys estimates from the sperm typing, and hopefully you can see there's pretty good concordance between these two. So, and we've got these kinds of data for actually 2 regions in the human genome, and they both show very good concordance apart from 1 hotspot. So that's at the very fine scale. We can also look at concordance with genetic maps estimated not at the very fine scale but a very broad scale, and in particular the genetic maps that people like Decode or Genathon or the Marshfield genetic map, those very large scale pedigree-based maps give resolution about variation in the recombination rate at the megabase kind of scale, and we see R-squares about 0.95 between our estimates at that scale, smooth to that scale, and these pedigree maps. So we believe that the results that we get out of this are sensible. So we can apply these methods across the whole genome. This is just chromosome 12. The black lines show you the estimated recombination rate. The red line, if you stare very hard, is the estimated recombination rate over the chromosome from the pedigree-based map. And we have a related technique, which I won't go into, for actually calling where hotspots are, which is what these dots are up there. Just ignore the bit at the top. So we've generated a map of hotspots across the human genome, and we've used it to learn lots of interesting things about recombination. Perhaps most excitingly is that we've taken the hotspots that we've identified by this method and asked, are there particular DNA sequences which are strongly enriched in hotspots and therefore are good candidates for actually causing the location of hotspots in particular places? And the answer is there does seem to be a very strong candidate for such a motif. This is the motif over here. It's lots of C's. It's rather degenerate. There are sort of subtle influences of genetic background, which is essentially what this picture shows. There's the motif stuck in the middle. This is essentially the p-value for how important influences either side of this middle 13 bases are, and you can see there are some additional strong influences outside. So we've gone a reasonable way to identifying a motif that strongly influences hotspot activity. We think it explains about 40% of all hotspots. And we have pretty good evidence that this thing really is causative, because in, I think now 5 hotspots that Alec Jeffreys has studied, actually humans are polymorphic for hotspot activity. So some people have a hotspot, some people don't have a hotspot. You can look at the DNA of those individuals that have the hotspot and those that don't, and as I said, in 5 out of 6 cases, the people that don't have a hotspot have a mutation in this motif. Okay.
How predictive is it across the genome? If you run, do you know what I mean? There's
Yeah.
a kind of false positive, false negative.
Yeah, totally. So, as I said, that varies enormously by background. So, there are lots of ways you can try and measure it. And actually, quite a useful way of summarizing it is to think of it in terms of a relative risk. What's the relative risk of having a hotspot given this motif to not? And if I just told you this motif is here, it's not high, it's like 3 or something like that. So it's good, but it's not great. On certain backgrounds, it's almost completely predictive.
that's, I mean, by background you mean the context. Of
Yeah, so
being it.
exactly. So in particular, if you have this motif or a variant of it in 4 repeat elements, that's the 2 THEs, line 2s, and
I see.
alu-Ys, then it's between 40, for the less hot of those, it's about 40% predictive, and the hottest is about 80% predictive. Okay. But just this on its own isn't. We think this on its own is useful, but it does vary a lot between background. There's clearly a lot else going on. We haven't solved the problem, but
Is there any relation between, I mean, so in some sense, the background plus this is the predictor.
absolutely, yes.
Is there any variation between populations in this?
Interesting question, and I don't have a good answer to that. It's actually very hard to get a good sense of differences between populations in terms of recombination rate. And the main reason for that is that human populations are pretty similar on the whole. Okay, so the differences between European population and African population are differences in frequency, typically not presence-absence of particular variants. And so what we might be looking for are differences in the intensity of hotspots between populations. So we can obviously take our estimates and say, do we see more or less combination in our population? But obviously you'd want to ask, do we think these differences are real, or is it just noise? And to answer that question, you have to have a good model of human history. And I'm not confident enough that I can fully capture the key, all the features of human history which are important in that. I mean, it's sort of on the list of things to do, but I worry about it quite considerably. Prof. Sorry.
Do you find relationships between your hotspot positions and where the genes are, or the genes do?
Yes, if I gave you if I'd given this talk all about recombination, I would have gone on to show you things like that. So if you look, these things don't particularly cluster around genes. If you look at, let's call this the transcription start site, what does recombination do around there? Well, what it seems to do is it peaks in the 5' region, which is not too surprising because open chromatin we know is very important for hotspot activity. But the interesting thing is that actually within the transcription unit, there's quite a big dip in recombination activity. And then if you look at the 3' end, no, it doesn't go up as high as that. It's sort of got a long shoulder. Don't really, and we can sort of make sense of this. This don't really know what to make of it. It's a real phenomenon. Basically certain it's a real phenomenon because the recent paper from Molly Chavorsky, which was analysing crossover events in pedigrees, found the same thing. So pretty confident that it's real, but don't really understand it. Yeah.
You are making the assertion, if I hear you right, that if you like, you could be a high recombination person and I could be a low recombination person in some sense.
Yeah. So that's certainly true at a particular point on the genome. There are also differences between people in the total amount of recombination they have. So there's polymorphism in hotspots, but it might well be that if you don't have that hotspot, you have one over here that I don't have. So there could be a compensation. That we know occurs to some extent. For example, in yeast, if you knock out a hotspot here, typically another one shoots up nearby. But then there is a different question of whether the overall amount of recombination that you have is different from me. There's definitely variation between people in the total amount. It's much more marked in women than in men, interestingly, for reasons that no one really understands. It may be linked to fertility. There are very strong indications that it's linked to fertility because you need recombination for appropriate segregation at meiosis. Okay. Any more questions on this before I move on and away from recombination?
So just to clarify, do you see that sequence motif trending with particular repeat families?
Yes, absolutely. So there are some repeat families like L1s which are very cold, okay, and you don't really see this motif in. There are some other repeat families, say like alus, where the vast majority of repeats don't have this motif. It's a real subset that do. And it's only those that do have this motif which have an increased recombination rate. And the frequency of this motif varies a lot between different repeat families, and actually the activity of those repeats that carry that motif varies between families. So, as I said, on a particular background, the THE elements, if you get this motif, you almost always have a hot spot. In fact, we think you always have a hot spot.
How ancient is that repeat family?
Ancient. It's about 50 million years.
Do we find different types of hotspots, or are they all the same?
How might they be different? So, I suppose the only... In yeast, for example, people have tried to classify hotspots into different types. And one of the key types in yeast that people claim is that a lot of them are associated with promoters. And we do see some. Clearly, we do see an enrichment of recombination around promoters, and so you might think that that's a class of hotspot, but actually the vast majority of hotspots are nowhere near genes in humans, so you might think they're slightly different. So we haven't actually separated whether this motif is the same at promoters, works the same at promoters and away from promoters. That would be an interesting thing to do. It's worth doing. It's very C-rich, as you can see, and we know that promoters are GC-rich. Okay, so I'm going to spend the last 20 minutes talking about something slightly different, which is, but also related to this notion of breaking up your data into small sets and combining inferences, which is the question of estimating local genealogies, and motivated by this picture. This is a 1 megabase region from a European population. You can see that across this megabase region there's essentially a haplotype that dominates. It's very, very extended, and people won't get too many prizes if they know anything about humans for guessing that this is the lactase region. Now, if we the goal of what I want to talk about is to say, take this region, point at a particular position, estimate the tree, and then use this estimate of the tree to tell us things about the age of the mutation, tell us things about population founding, tell us things about migration and admixture in a sort of more general sense. So I can do that. I can do any of those things anywhere across the genome. So, sort of generally, estimating genealogies at any position along the genome can be very useful for learning about diverse processes, including these. And the insight I want to try and pursue is that if we're trying to learn about the tree, say at this position, we get a lot of information by essentially comparing the haplotypes at that position. If we have a very long haplotype like this, individuals that are identical over the whole region are clearly going to be very closely related, whereas individuals which, say one's here with one's here, there's been a breakdown in this haplotype sharing, which suggests that they're not quite as closely related. So more generally, this breakdown in haplotype sharing is going to be informative about the tree at this central position. And you can, where I guess the key insight is that very shallow parts of the tree, i.e. where there's a very recent common ancestor, are going to persist, are going to lead to haplotype patterns persisting over the whole region, whereas much deeper parts of the tree are only going to persist for very short regions. And so if I have a tree at any particular position, I can think of how much of this tree is maintained as I look to the left and the right of that position. So what I'm going to show you is a movie that is going to move away from a particular position and see how much of this tree persists as I move away from it. And what you can see is that very quickly a lot of that tree breaks down, but then some parts of it persist for large genetic distances. So this is a scale in genetic distance. So there's a clade of this tree which is going to actually persist pretty much to the end, whereas the deep structure tends to break down very quickly, whereas the tip structure can last for a long while. So what we'd like to do then, here's the sort of the broad setting. We'd like to take these data, learn about the tree, and just as we took subsamples of size 2 in the case of the estimation of recombination rate, we can also imagine taking subsamples of size 2 from our sets of haplotypes to learn about how deep the tree is. So here again, 2 haplotypes. I've just used noughts and ones this time instead of lines or blobs. Yet another notation, apologies about that. The red numbers indicate differences between these sequences. You can see there's a large region where they're identical, so we think there should be a shallow tree here. There's a region here where they're really quite different. We think there's a deep tree. There's a region over here where they're a little bit different, and so we think there might be a semi-deep tree. And so what we can do is, something really rather simple, is that we can try and estimate what's the distribution of trees for a subsample of size 2 at a particular point on this chromosome. But what we've got to allow ourselves to consider is the fact that the depth of this tree varies along the sequence. And so essentially what we want to do is to get effectively a posterior distribution on the tree depth at this position, and to do that we can use we can combine hidden Markov model structures with numerical techniques, like particularly Gaussian quadrature, to estimate actually not the posterior distribution, but the marginal likelihood surface at a given position x. So I want to calculate what's the probability that I've observed these 2 haplotypes, given that the time to the most recent common ancestor at this position takes some value. And I can do that, I can get these surfaces using these numerical techniques in just the same sort of way that I was doing in the recombination case. I'm going to take a little bit of the data and I'm going to calculate a likelihood surface. I'm actually going to take a further approximation, which is I'm going to take that likelihood surface and approximate it by something else. In particular, I'm going to take this approximate likelihood surface from a discretized space, and I'm going to fit a continuous model using a scaled gamma distribution. The reason I do this is that this massively reduces the computational load in further steps, and actually it's reasonably justified, because in the case of there being no recombination, actually the truth is a scaled gamma distribution. So I'm going to take this sort of approximate surface and I'm going to approximate it even further using, so I essentially just get 3 numbers, which is the 2 parameters of the gamma distribution and some scaling factor. And that gives me an approximate likelihood surface. Okay, so what am I going to do with these likelihood surfaces? Well, I'm going to use them to build a tree. So imagine we have a partially reconstructed tree. So I've got 10 sequences here, and I've reconstructed the tree up until some point. And what I want to do is to try and reconstruct the next step in that tree. How would I go about doing that? Well, what we can do is to approximate the probability of any further step in the tree using the composite likelihood. So I could imagine saying that the next step is a coalescent between this clade and the one by it, and that these 2 groups coalesce at some point t, and I'm going to call these 2 groups alpha and beta. I'm going to say that the probability of this is proportional to the product over all chromosomes in I and all chromosomes in beta of the marginal probability of observing these 2 chromosomes given this time. And just as I was doing in the recombination case of saying how bad can this approximation be, so that I end up with something that's a bit more like a likelihood. I do the same thing here. I rescale, so I reduce the double counting. And so I've got an approximation for the probability of this using my composite likelihood. A sort of a technical note, which is that effectively what I'm doing is assuming that these uncoalesced ancestors are independent draws from a stationary distribution, which is something I may want to revise later. Now there's an important detail actually. So I'm going to use the composite likelihood to calculate the probability of further steps in the tree. You could imagine considering every possible further step in the tree and then choosing among those in some way. I'm actually not going to do that exactly. I'm going to use a slightly different construction, which I refer to as the nearest neighbor construction. The reason for doing this is that it gets over certain biases that the alternative construction doesn't. The alternative construction has. So in this nearest neighbor construction, which is actually just a reconstruction of the coalescent process, which is why I use it, the idea is that each lineage chooses a nearest neighbor. So each of these lineages here are going to choose who they're closest to. Then I'm going to choose which of these nearest events is the next one to happen, and then I'm going to choose a time for that. And that splitting it into those 3 steps actually enables me to get around some of the inherent biases in considering all possible ones. I mean, it does consider all possible ones, it just rescales them in a slightly different way. But the key point is it still uses the composite likelihood. What then do we do? So what we've done then is given ourselves a way of evaluating different further steps in the tree building process. So we can use these functions to then choose the next event, and you might want to maximize, or you could sample from some pseudo-posterior. And this is essentially where that gamma approximation comes in, because if you use this gamma approximation, this leads to a highly efficient algorithm for estimating or sampling the local genealogy that has the same time and memory complexity of neighbor joining. And we know that neighbor joining can be applied to massive data sets, and so this means that this should be able to be applied to massive data sets too. So again, I'm really being motivated here by the notion that we want to apply these things to large data sets. What does the algorithm do? Is it a good algorithm? Well, you've got to try and decide what are good properties for any such algorithm to estimate local trees along a genealogy. And here are some things that properties that this algorithm has, which I think are desirable. It can be fully stochastic, unlike obviously things like neighbor joining, UPGMA, or maximum likelihood. It returns the prior in the absence of data, seems fairly sensible. It returns the truth in the limit of infinite data, also seems sensible. It's true under it's correct under certain limits. For example, if our data set consisted of a single SNP, and we thought the mutation rate was very low, it does the correct thing. It's very close to the optimal proposal distribution in the case of no recombination, which again also seems sensible. It uses, well, I don't think I should allow myself to use much. It uses some available information, and it's fast. So here's just a toy example. It's actually I've given myself a very hard toy example here for some reason. There's a true tree at some position. Here are 6 draws from the pseudo-posterior for what the tree looks like. I won't go into huge detail about it, but it sort of gets some bits of it right most of the time, and other bits it does fairly poorly on. But I've actually given myself a very hard setting here where the mutation rate and the recombination rate are the same, so there's actually not much information at the tree in that setting. So we'd like to be able to, so we can obviously estimate trees or sample trees using this. How do we evaluate tree accuracy? Well, that clearly is going to depend on what you want to do with these trees at the end of the day, and specific applications will require some bits of the tree to be more accurate than others. However, there must be some sort of sensible way of comparing two trees without being too specific about what you're going to do, it seemed to me that a sensible thing to do is to compare the representation of bipartitions in the true tree to the estimated one. So you can think of a tree as inducing a whole bunch of bipartitions on the sets of sequences, and the similarity between an estimated tree and a true tree could be measured by looking at these bipartitions. And we're not actually going to require exact matching bipartitions. We're going to essentially look at a correlation between bipartitions induced, or a maximum bipartition, maximum correlation. And we're going to weight by branch length. Sorry, this is probably too much detail, but the key point is that if you just take your data and you do something that's not totally daft, for example, you just applied a clustering algorithm and you looked at the dendrogram that came out of that and you asked how well did that represent the tree, this is just a single sample, you can actually do reasonably well with an average weighted maximum R squared of about 0.6 in terms of these bipartitions. But if you do this more sensible thing, what I think is a more sensible thing of trying to take samples from the pseudo posterior, your average weighted maximum R squared for these bipartitions is very good, and this is still quite a difficult situation that I've given myself. Okay. So, but it's worth saying that this is still very much work in progress. It's not. I wouldn't say I've got full answers to everything. Okay, so what I hope to have convinced you of, I think, is that although composite likelihood, this idea of breaking your data down into little bits, performing inference in a sort of full way on those little bits, and then combining the data in semi-sensible ways, although it breaks many fundamental rules in statistics because of assuming non-independence where there's non-independence, it nevertheless can lead to useful techniques, at least in the two settings that I've described. And I think that there are many settings in statistics where composite likelihood has been useful. Nevertheless, there do remain very important and rather open questions in these applications, and more generally. There's this issue which I've referred to a few times of obtaining useful estimates of uncertainty. As I said, you can do power transformations of composite likelihoods, which can sort of bound how bad things are ever. But you'd actually like to like to have well calibrated pseudo posteriors, and so could you actually do better? Could there be adaptive methods, for example, to try and improve the ability to get useful estimates of uncertainty? Then there's another question which I think applies particularly in the latter part of my talk, this estimating genealogies, is that rather than using subsamples of size 2, can we use larger subsets of the data, for example quartets, to learn about these quartets and then combine that information sensibly? And that would be a very useful thing to do. It's actually been tried for the recombination rate estimation question. People have tried using sets of 3 SNPs for the recombination rate estimation. And it actually doesn't do any better than using subsamples of size 2. So it's not always doesn't seem to always massively improve your inferences to use larger subsets. But I think in the genealogy estimation setting, it would be a major improvement. Then just some acknowledgements. Many thanks to people in Oxford Statistics. The recombination work was very much done in collaboration with Simon Myers. Chris Holdsworth is working on the genealogy. Adam also worked on recombination, as did Colin and Peter Donnelly. Paul Fernhead helped in the early stages of the recombination work. And many thanks to people in the HapMap project for a good time. So, thank you. Any questions? Kept you a long time.
Is there any value in thinking about, in the sampling process, rather than saying using quartets, working out which SNPs are really redundant, which SNPs would truly be double counts, and which pairs of SNPs or which set of SNPs are sort of representing the information.
Yeah.
I don't.
No,
There's something intuitive about that, but I don't know.
That is sensible. So there's another composite likelihood method for estimating recombination rates, and actually variable recombination rates, that Paul Fernhead developed, which, rather than taking all these pairs, it cuts the data up into little chunks, okay, and then within each chunk it throws away SNPs that it thinks aren't informative, and then it estimates the recombination rate for that little chunk, assuming a constant rate. So it's and that does reasonably well. It doesn't scale very well. The problem is even small chunks are very, very challenging. But I certainly think, certainly in the recombination, there are SNPs which are totally uninformative and ones which are very informative. And there are things you could do to speed it up.
Two questions. I guess the first is, so you're using the pseudo likelihood inside the reversible jump?
That's right, yes.
Do you have any handle on how much bias that's introducing into model probabilities by using it?
Well, it's going to have the same effect of being over certain. There's no doubt about that. As I said, we have explored ways of correcting that over certainty by doing these power transformations. And when you take a worst case power transformation and you look at the you look at the estimates of uncertain, how well calibrated the posterior is, and that it actually that does a reasonably good job. So, well, when I say reasonably, I mean it's not perfectly calibrated, but when I say 95%, it may be.
Okay. So the second question is sort of in line with that, and maybe it's something for more of a coffee, but it seems like if you specified a conjugate prior over the thetas, then estimating model probabilities would actually come down to ratios of normalizing constants. And granted, the integral there would be impossible to compute, but the Laplace approximation only requires the mode of the distribution, and it seems like the pseudo likelihood does very well at hitting that. So it might be an easier way to get model probabilities.
Yeah, okay. That's an interesting idea. Talk to you about it.
I know, about not having to use reversible jump, you could use MC3 or stochastic search to actually search the model space instead. It might be a lot quicker.
Yes, there are many alternatives for exploring model space. For example, people have tried discretizing using hidden Markov models. Speed isn't actually an issue at the moment. This actually works really, really well. So in terms of speed, so
convergence issues at all. Sometimes when we get into high dimensions, we find reversible jump just takes a while.
yes, this seems to be one of those cases where reversible jump actually seems to work remarkably well. So I agree, there are many settings where reversible jump does stick, but this doesn't seem to be one.
Since I'm working in this area at all, I'm just ask a naive question. Where are the priors here?
We're talking about the recombination.
Well, first of all, the coalescence itself is presumably an approximation.
Yes.
And to whatever's happening, and it is a parametric model.
Of course. Yep.
And so, where do the priors on the parameters come from?
Okay. So the coalescent is itself a prior, obviously, on the genealogies. So that's a broad sense. Obviously, I want to specify priors on these recombination maps as well. That's that's a sort of thornier. Well, you can imagine being more data-driven in that. So in what I've described, we originally used a rather unrealistic prior because we didn't know much about what recombination looked like. So we just had changes in rate as a exponential process, Poisson process, homogeneous Poisson process with rates in those drawn from an exponential distribution. We now know that the world is hotspotty with large regions of low recombination between, and a student of mine did a project where he actually had a prior that he fitted a model where we were sampling over hotspots and background rates, so we changed the prior, and we were very much driven by what we'd learned about to start with. So I think I mean, it's clearly an important issue. The inferences about recombination, I tend to prefer fitting less data-driven priors, and so I quite like the one that we fitted in initially because it's very unrealistic. So if the data's strong, then you can tell when the data's strong, I suppose.
I'll just ask about another. Do you have any sort of asymptotic theory, if you want, apart from, of course, that, I mean, certainly if you have a lot of individuals, and eventually, of course, even then, you still, even if you have the whole population, you're not in the kind of regimes that one is with.
Yeah.
But are there sort of regimes where you can get simple approximations to estimates of parameters that where you can fit reasonably well?
I haven't looked at that personally. Paul Fernhead's done a reasonable amount of work on that. In the recombination rate estimation, the method that I've described, at least in the way I've implemented it, is consistent in the sense that even though you have this double counting, if you have enough data, you get the answer right, as is his method. The original method that Dick Hudson proposed isn't, because of the way in which you break the data up. So, and I suppose in the second half, you know, some of the limits that I was talking about are, in a sense, that's sort of what you're aiming for. Those are the limits that I thought we should focus on. So, I mean, it's worth saying, actually, I'm not a statistician. I'm a biologist by training, and some of these questions, which are clearly very important, it would be probably do need more attention, I think.
Going back to the question of determining the subset size, would it make sense to use data-driven methods to see how different blocks of data are? Like something, for example, in the direction of yesterday, that we want to determine the blocks of.
Yeah, I agree with that. I mean, I think there's all sorts of things you could do about using block sizes, you know, to improve your block size choice. Everything I've spoken about, you know, was working with block sizes of 2, and I haven't got anything that works with block sizes of more than 2. So at this point in time, I'm not in a position to explore that. I think I think the next step is to just try increasing the size of the subsets, and then after that, maybe thinking about adaptive methods.
Has anyone looked at the correlation between the hotspots and DNA's hypersensitive sites?
So yes, we have. There's none, basically, which is slightly worrying, because we know in yeast that all hotspots are DNase 1 hypersensitive. So why don't we see correlation? It's probably because you know, no one's measured DNase hypersensitivity in a meiotic cell line. There's no meiotic cell line available for mammalian work. So we just don't think we've got the right information for that. So.
People done it in sperm now?
That's a really interesting question. I don't know. No is the answer, but it presumably could be then. Yeah, that's a really nice idea. Richard.
So I'm so interested in your way of reconstructing at the end with the when you were building the genealogy. I guess I just wanted to kind of so you, for each pair of, at some point you've reached, for each pair you evaluate this posterior gamma function, and you draw from it, you take the most, the next one, the shortest event that you
You can either I sample.
sample.
So you could maximize, but I would prefer to sample.
And what about mutations?
I don't represent mutations at all.
Do you sort of absorb them in a greedy way, but or because that's going to affect whether things
So I only use the mutations once, which is in estimating these surfaces. I never look at them again. So the trees that I build will not necessarily, suppose I believed in infinite sites, the trees I build will not necessarily fit the infinite sites model. Okay. Now, there's another way of constructing them that uses these marginal pairwise things, that does very specifically. So here I've said that each of these are independent draws from a stationary distribution, but if you actually link them together by a star and you use that approximation, that, funnily enough, would recover the infinite sites part in the case of no recombination. So there are different choices you can make here, and none of them is ideal. For example, if you put the star in, it leads to other biases, which I'd rather not have, which is why I don't do it. And this will typically lead to trees that are concordant with infinite sites, unless you've got very strong heterogeneity in the rate of mutation down branches. But you know, there's a bigger question as to what you want these trees to do, and if you want them to, you know, if you care very much about them agreeing with infinite sites, then you go down one path, and if you don't care about that so much, you go down another path. So I mean, it's just a question of what you want these to do.
Infinite sites isn't completely correct.
No, exactly, and that's why I don't want to put too much weight on it.
All right, so I think we'll hold off any other questions for the coffee break, and let's thank Gil again.